So B cells are produced in the bone marrow from stem cells. So stem cells are the origin of all types of blood cells. So you have red blood cells and then you have white blood cells. And all of those are going to arise from bone marrow stem cells. And there are many types of B cells that are produced. So what I mean by types of B cells is that they're going to have different receptors. So different B cell receptors. BCR is short for B cell receptors. So let's say there is one B cell which is going to have a receptor that is going to be shaped like a semicircle, and then another B cell with a receptor that is shaped sort of like a triangular shape. And note that these B cell receptors are actually antibodies. So B cells are going to have antibody receptors, and each type of B cell is going to have a different type of receptor. These different types of receptors are going to refer to different types of antigens. So for example, the triangular shaped receptors are going to bind to triangular shaped antigens, and the circular shaped receptors are going to bind to the circular shaped antigens. So these are antigens. Now, what do I mean by an antigen? So an antigen is anything that will lead to an immune response. An immune response is normally against pathogens or against infected cells, right? So let's say we have a bacteria over here, and this bacteria has these substances on its cell membrane, which give it its identity. So what that means is that these surface molecules are going to be different for different types of pathogens. So let's say this is bacteria A, and then we have another bacteria B. And this bacteria, how do we differentiate between bacteria A and bacteria B within the body? The body differentiates them based on the type of antigen that they are going to produce. So bacteria B has this triangular shaped antigen, and bacteria A has these circular antigens. So this allows these different shapes or different types of antigens on different types of bacteria are going to enable us to distinguish between them. And that will allow different B cells to identify different types of antigens. There are many thousands of B cells within our body, each with a different type of B cell receptor. And this enables us to identify a whole range of pathogens, to identify a range of pathogens. So we have the potential, our immune system has the potential to identify a variety of pathogens and then initiate immune responses to help us fight against them. Now, as for B cells, the way B cells act is by producing antibodies. However, B cells normally cannot produce antibodies because they're in the naive state or they are in the immature state. And we call that immature or naive B cell. That's what we have normally in the blood and in the lymph nodes. So if you don't know, the lymph nodes is actually the organ where the B cells and T cells are going to be in the highest concentration. So you can find the B cells in the blood or in the lymph node. You can also find them in the spleen. These naive or immature B cells are what we have when our body is in a normal state and not infected by any pathogens. However, when we are going to be infected by pathogens, these B cells are going to go through a number of changes upon infection. And the B cells are going to be converted into plasma cells. These plasma cells are going to secrete antibodies. And these antibodies are going to help them fight the infection. And then when the infection is over, the plasma cells are going to die, and we're going to be left with memory cells. So the plasma cells and memory cells are going to be formed at the time of infection from immature B cells. And then after infection, plasma cells are going to die because they have done their job, and the memory cells are going to remain in the blood. And they're going to remain just in case the pathogen infects again. Then these memory cells, which have receptors that are specific to a particular pathogen, can act against that particular pathogen. We're going to look at more of that in the secondary immune response. Now here we said that immature B cells are going to be converted through some steps into plasma cells and memory cells. Now there are two ways in which a naive B cell can be converted into plasma cells. So there are two ways in which plasma cells can be formed. And note that I'm only saying plasma cells and not memory cells, because one of the ways is going to produce both plasma cells and memory cells, and the other method is only going to produce plasma cells and not memory cells. So what are the two ways to produce plasma cells from B cells? The two ways are, number one, T-dependent antigens, and number two, T-independent antigens. Now, we're going to look at each of these in detail. First, we're going to look at T-dependent antigens. So what the term T-dependent antigen refers to is that these are basically antigens that will activate an immature B cell to produce plasma cells. However, that activation will involve a T helper cell. So that's why T-dependent antigens. So we're going to have these soluble antigens in blood, which basically means that they can be dissolved in blood. The soluble antigens could refer to these substances like peptides that are produced by, for example, bacteria, or they could refer to the bacteria themselves. These soluble antigens are going to travel through blood and they're going to go to the lymph nodes. The lymph node is an organ where you have a very high concentration of B cells and T cells. So you can say that this is the B cell and T cell home. You have a lot of B and T cells at the lymph node. So what's going to happen at the lymph node is that these soluble antigens are going to bind to the cell membrane of macrophages. And then we're going to have something like this with a macrophage that has antigens all over its cell membrane. This macrophage, which is bound to an antigen or antigens, because there are many, is going to be called an antigen presenting cell. So APC, because this macrophage is presenting antigens on its cell membrane. The purpose of this antigen presentation is that now these antigens can be bound to B cells. Remember that B cells had antibody receptors for binding to antigens. And also recall that these B cells are of different types, right? So there are B cells that can bind to, say, circular antigens, and there can be B cells that can bind to, say, these triangular shaped antigens, right? So note that I'm giving these random shapes just for understanding. They're not really actual shapes for antigens. So in this case, we have a circular antigen, so we're going to have this type of B cell that can bind to these circular antigens, and the other B cells that cannot bind to circular antigens are not going to bind to this macrophage. Only the cells that have receptors for this specific antigen will bind to this macrophage. What's going to happen next is after a B cell binds to this macrophage in this way, it's going to take in this antigen. So what that implies is this B cell is going to engulf this antigen. So it's basically going to, I'm just going to show an enlarged portion. So this is just the B cell in a larger view. It's going to capture that antigen within a phagosome. So it's going to capture that antigen within a vesicle or a phagosome, which is basically which is basically a vesicle that forms by phagocytosis. So we have an antigen over here, and this is the phagosome, and this is the B cell, this is the nucleus. What it's then going to do is a lysosome is going to come and bind to this, right? So we have a lysosome is going to come and bind. So these are the enzymes within the lysosome, right? This lysosome is going to come and fuse with this phagosome, and eventually these enzymes are going to affect this antigen in some way and process it eventually. And this antigen is going to be processed by the cell in order to eventually express it on an MHC2 molecule. So this is an MHC2 molecule. Now, this might seem a bit complicated, but bear with me. So an MHC molecule is something that is used for antigen presentation. In this case, we have the class 2 MHC molecule, and that's because the class 2 MHC molecules are going to attract T helper cells to come and bind to it. And why we need T helper cells is because without T helper cells, these T-dependent antigens cannot really activate the processes that are going to be required to convert this immature B cell into plasma cells that can produce antibodies. So remember that this stage is going to be called antigen processing. And we're going to have a number of things that are going to happen here, like this antigen is going to be processed, it's going to be attached to an MHC2 molecule that is going to be produced inside the cell. And eventually, that MHC2 molecule and antigen is going to be expressed in the cell membrane, like this. Okay. What's going to happen next is that a T helper cell is going to come in. TH stands for T helper cell, is going to come in and is going to bind, is going to have a receptor that is going to be able to bind to this antigen. So an MHC2 molecule will help hold this antigen on the surface of the B cell so that it can be recognized and bound to by a T helper cell. We're going to look at more about T cells and these MHC2 molecules in the T cell video, but just know that T helper cells are going to be activated by, T helper cells can bind to cells that present antigens using the MHC2 molecule. Now, why do we require binding of the T helper cell? That's because the T helper cells are going to secrete certain molecules called cytokines. So cytokines are basically hormones that will help activate the B cell. The cytokines will activate the B cell. And of course, the B cell is going to have cytokine receptors at this point. So it's going to receive those cytokine molecules and then get activated. What I mean by B cell activation is that the B cell is eventually, once again, there are a number of steps that happen during this activation stage. And the B cells are going to be eventually forming plasma cells. And memory cells. Plasma cells, remember, plasma cells are cells that can produce antibodies. So obviously they're still going to have those antibody receptors, but they're also going to be able to produce antibodies that can be secreted into the blood, for example, so that they can do their variety of functions like agglutination and neutralization of toxins, etc. And memory cells are cells that are basically going to remain in the blood. So they're going to remain in the bloodstream and they're going to keep circulating until you encounter the same pathogen a second time. So that is the entire process for forming plasma cells from immature B cells through the T-dependent antigens. Now I'm going to give a star over the plasma cells over here because we're going to return, there's something special about these plasma cells compared to the plasma cells that are produced from the T-independent antigens. And I'm going to go through that towards the end of this video. So now let's look at the second way, which is T-independent antigens. So this is a simpler procedure because we don't require T cells, so it's much simpler. However, it's not going to be as effective as we'll see. So in the case of T-independent antigens, we're going to have these antigens. Now these are larger antigens compared to the T-dependent antigens. These larger antigens are not going to bind to B cell receptors. So they don't bind to B cell receptors. So remember that in the T-dependent antigens, these were the B cell receptors, right? These were the B cell receptors, the specific antibody B cell receptors that bound to the antigens. However, it's not going to be the case in the case of T-independent antigens. So in T-independent antigens, it's not going to bind to B cell receptors. Rather, it's going to bind to other receptors, for example, the toll-like receptors, etc. So it's going to bind to some other receptors that are going to look a bit different. So this is just an example, not a very accurate representation of the actual receptor, but it's going to bind to other receptors rather than so what I mean is this is a B cell receptor and this is another category of receptors. So it's not going to bind to the B cell receptor, these antigens, rather they're going to bind to other receptors that can be found in the B cell, and that's for T-independent antigens. So this binding to other B cell receptors is going to activate the B cell directly. So there is a direct activation. You don't need the involvement of T cells. No T cells are needed. To be more specific, no T helper cells are needed for activation. What this is going to produce is plasma cells only. So T-independent antigens can activate B cells and produce plasma cells, and there are not going to be any memory cells. So there won't be any memory cells. So that's a disadvantage, right? Because if there are no memory cells, this means that there is no secondary immune response. No secondary immune response because you don't have memory cells. So if you are infected by a second time by the pathogen, then you're going to have to go through the entire procedure once again. And that's an disadvantage. And it's only going to produce plasma cells. Now I'm going to talk about the plasma cell differences, which I mentioned, right? So I said that there was something special about these plasma cells compared to the plasma cells produced by T-independent antigen activation of B cells. And the difference is the plasma cells which are going to be produced by the help of the helper cells, these plasma cells are going to be long-lived. So this means that they're going to have a longer life. They will be able to act against the pathogen for a longer period of time. And they're also going to be more specific. And that means they can provide a better immune response. They can provide a better immune response. So what I mean by more specific is that the antibodies that they are going to produce are going to have a higher affinity for the antigens of the pathogen, So if they have a higher affinity for the antigens of the pathogen, then that would mean that they will be able to bind to the pathogen antigens better, so they can perform the antibody functions better than these plasma cells, which are only going to produce low affinity antibodies. And they're also going to be producing this for a shorter time because they have a shorter lifespan. So they die quicker than these plasma cells. And that was all for B cell activation. So just for a recap, there are two ways in which you can activate B cells. You have T-dependent antigens and T-independent antigens. When we're gonna take a look at the dependent antigens, we start with soluble antigens that can travel through blood to the lymph node, where we have a high concentration of B and T cells. B cells, so these soluble antigens are going to bind to macrophages and be expressed on their cell membrane. And then they are going to be binding with B cells that have specific B cell receptors for that particular antigen. That antigen is then going to be engulfed by the B cell, which can then process that antigen to express it on its cell membrane so that a T helper cell can come and bind to it and eventually produce plasma cells and memory cells. And then the other way is through T-independent antigens, where we don't have T cells involved in the process. It's a direct activation. So antigens are going to come and directly bind to the B cell, and there are not going to be any T helper cells, and it's going to produce plasma cells, which are low affinity antibodies with a shorter lifespan. And this is not going to have any memory, so it won't be able to produce a secondary immune response. And that's all for B cell activation.